Hello. In this brief presentation, I will give a general overview of how exposure to traumatic experiences can affect brain function and behavior. When we talk about trauma, the first thing to mention is that indeed, from a neuropsychiatric point of view, exposure to traumatic experiences such as shocking, scary, or dangerous events can lead to PTSD or post-traumatic stress disorder. And we also know that the main symptoms of PTSD are experiencing and avoidant behaviors, enhanced arousal, cognitive alterations, and reduced mood. In addition, being exposed to traumatic experiences can lead to several other behavioral changes, particularly when exposure takes place during early development. Our lab has contributed to establish a causal link between exposure to trauma, specifically during the peripubertal period. This period comprises childhood and adolescence, and the subsequent risk to develop memory and attention deficits, anxiety, helplessness, depression, reduced sociability, and increased aggression. As we will see, there are important differences in the susceptibility of individuals to stress and trauma, and understanding the reasons for these differences and the underlying neurobiological mechanisms are one of the main lines of research in our lab. A main reason why the peripubertal period is an age of particular vulnerability to the long-term effects of traumatic experiences is that during this period, the brain is still developing and shaping its connectivity, something that takes place until well-advanced adolescence. I will now illustrate some of the key findings obtained in our lab with regards to the programming of adult psychopathologies following traumatic experiences during peripuberty, with a particular focus on aggression. We know from epidemiological and clinical studies that being exposed to a high stress or violence during childhood is the highest risk factor for the development of high aggressive or violent temperament as adult, something that is known as the cycle of interpersonal violence. In our lab, we have used rodent models that are devoid of societal or cultural influences, and this allows us to reveal important biological factors that mediate the long-term changes in aggressive behavior, including epigenetic changes that are triggered by exposure to traumatic experiences during early life. In addition, using a long range of molecular, histochemical, and genetic techniques, we could establish also that traumatic experiences change the natural balance that exists in the brain between excitation and inhibition. And we could identify some of the key molecules, including neuroligin 2 from the cell adhesion molecules family, as key players of the cognitive changes induced by peripubertal stress, and therefore as potential therapeutic targets. Furthermore, we have revealed changes in glucocorticoid receptor expression in the brain taking place in peripubertally stressed individuals, and subsequently we performed pharmacological studies in which we show that treating with a glucocorticoid receptor targeting drug at adulthood effectively reverses pathological aggression in individuals that had experienced peripubertal stress. In another study, using magnetic resonance and diffusion tensor imaging, we identified microstructural changes in brain regions involved in cognitive function and decision-making that were specifically observed in the subset of individuals that develop pathological aggression following peripubertal trauma, not in the ones that don't develop those behavioral changes. In addition to studying the impact of trauma during development, our lab is also concerned with the understanding of how exposure to stressors or being chronically stressed at adulthood affects our behavior and impacts our brain. Work from many labs worldwide, including ours, has shown that chronic stress can modify the structure and functioning of brain circuits involved in emotional processing and cognition, leading in many individuals to impaired learning and memory capacities and to a number of psychopathologies. Strong current focus in our lab is the study of the role of mitochondria in behavioral changes induced by stress and trauma. Mitochondria are small organelles, the powerhouses of cells, and they exert many different biological functions. They are expressed in high number in neurons, as shown here on the right in this electron microscopy image of a part of the brain. One of the main reasons why mitochondria are postulated to be key to mental health is the fact that the brain consumes almost 10 times more energy than the average of the body. And therefore, under conditions of compromised mitochondria, before than any other body organ, it's the brain that shows particular vulnerability to damage. We have recently shown that chronic stress in rodents and major depression disorder in humans leads to alterations in the expression levels of a number of mitochondrial genes in the prefrontal cortex. And these changes are observed in the subset of individuals that show high vulnerability to stress, those that develop particularly strong depressive symptoms. Furthermore, by applying neuroimaging and magnetic resonance spectroscopy in individuals that develop depressive behaviors following chronic exposure to stress, we have identified changes in the levels of specific metabolites in brain regions involved in mood and cognition, such as the nucleus accumbens, a part of the ventral striatum, and a brain hub for motivation. As shown in this slide, chronic stress led to a reduction in glutamine and in phosphocholine in the nucleus accumbens. And this type of data is allowing us to postulate the importance of certain nutritional interventions to target the chronic stress-related disorders. And that's something that we recently did in a study in the lab in which we gave a nutritional intervention, in particular L-acetylcarnitine, that was able to reverse depressive behaviors and metabolic changes induced by chronic stress. Lastly, I would like to mention a line of research in the lab in which we use virtual reality to acquire high-density behavioral data, physiological and electroencephalogram data as well, that combined with machine learning approaches is helping us to understand the factors that explain why different individuals have different susceptibility to stress. This is an example of an individual immersed in such virtual reality studies, and from here the next step is to develop bio and neurofeedback approaches that can couple with virtual reality to train participants to increase their resilience to stress. And I finish here this presentation about our research on the neurobiology of stress and trauma by thanking the multiple outstanding lab members that have contributed to the work over the years, to our collaborators and funding bodies, and to you for your interest. Thank you very much.